Welcome students, myself Dr. Barnawal. I am giving my lecture on the topic mustard diseases. Actually, there are so many diseases which is caused by fungi, bacteria. First of all, I am presenting the list of bacterial diseases and fungal diseases and causal origins of the pathogen. Now, coming to the bacterial diseases. Actually, there are three bacterial diseases. That is bacterial black rot caused by Xanthomonas campestris, PV campestris. Second one is bacterial soft rot that is caused by Erwinia cratofora. Earlier name was Pseudomonas marginalis, PV marginalis. Third one is Xanthomonas leafy spot that is caused by Xanthomonas campestris, PV arboraceae. Actually, these are the three important bacterial diseases of mustard, and 9 fungal diseases also. That is also very important for management point of view. So, first of all, that is Alternaria blight that is caused by Alternaria brassicae and Alternaria brassicicola. Second one is downy mildew that is caused by Pernospora parasitica. Likewise, Sclerotinia stem rot caused by Sclerotinia sclerotium serum. White rust that is caused by Albugo candida. Next, these 4 diseases, that is Alternaria blight, downy mildew and Sclerotinia stem rot and white rust. We will discuss in details about these 4 diseases. And rest disease, that is black leg caused by Leptospirea maculans, and the number of stage of the pathogen is Phoma lingam. That is, next one is Cercospora leaf spot caused by Cercospora brassicicola. A damping off that is caused by Fusarium species, and Rhizoctonia solani. Also, if pathogen is Rhizoctonia solani, then the polymorph stage of the pathogen will be Theratophorus cucumeris. Next one is powdery mildew that is caused by Irresipi polygony, and last one is white leafy spot that is caused by Pseudocercosporella capsicum. These are the list of fungal diseases. Now coming to the first disease, that is Alternaria blight of mustard. Actually, this is one of the most important diseases of mustard that causes heavy losses in all mustard growing area in India. This disease causes severe loss in Jharkhand state, and this disease is caused by Alternaria solani and Alternaria brassicae and Alternaria brassicola. These are the 2 pathogens. Generally, Alternaria brassicola, the pathogen are long, septate, and generally mutiform septa is there. In case of Alternaria brassicae, the spore length was small. In generally, as we are seeing in the slide also. Actually, when we talk about the classification of the pathogen, then the kingdom of the pathogen will be Mycota. Division is Eumycota, subdivision Tetromycotina, and class Hypomystis, order Moniliales, and family Dematiaceae. The characteristic feature of Dematiaceae is colored conidia, which is seen in the slide also. Colored conidia is the characteristic feature of Dematiaceae family. When we talk about Alternaria pathogen, we can say when we see under microscope, then definitely the tip of the conidia will look like a big sepet. Second one, colored conidia. Third one, muriform septa. Muriform septa means transverse septa and vertical septa. Both type of septa is present. That is called muriform septa. Muriform septa is the characteristic feature of Alternaria. So, in this way, we can say this is the characteristic feature of the Alternaria species. Next is symptoms. Now coming symptoms. Generally, the symptoms of the disease appears in leaf as well as stem. The disease attacks on the lower leaves as a small circular brown necrotic spots which slowly increase in size. As the disease advances, concentric rings are formed. Concentric ring means ring over ring. Generally, spots initially it was brown, and after that the ring over ring like structure that is called concentric ring, which is present in the leaf. And like many concentric ring, after some time, as the disease advances, after some time many concentric ring coalesces to form big patches and showing lightning and defoliation in severe cases. Circular to linear dark brown lesions also develop in the stem. After some times, the same spots also appear in pods also, which are elongated at later stage. Infected pods produce small discolor and shrivel seeds also seen. This is the characteristic feature of Alternaria blight of mustard, which is seen in leaves as well as stems. The photographs are clearly seen in the slides. Now come to the disease cycle. Actually, it is seed-borne disease. So, because seed-borne disease, when we sow the seeds, if seeds are infected, then definitely the symptoms of the disease appears first on leaf, then stems, and last at pods also. So, in first black spots appear in brown to black spots appear in leaf, and then comes to the stem and pods also. And infected pods, if sown, then this disease causes infection in plants also. Sometimes the pathogen also survive is debris also, crop debris also. So from crop debris or from infected seeds, pathogen blown away or you can say infect the healthy plants and symptoms appear. Then after some time, infected leaves, the pathogen blown away through air and infect the healthy plants. In this way, these cycles completed. So as well as survival and spread. Now survival and spread. The disease is externally and internally seed-borne. The pathogen survives through spores or mycelium in disease plant debris or weeds. The fungi is seed-borne and cause shriveling of seeds and low germination. Spores and mycelium in disease plant debris also serve as means of pollination. Conidia are abundantly formed in moist atmosphere and are disseminated by air currents. When we talk about favorable condition, then definitely we can say relativity play an important role, as well as temperature and rainfall also play an important role for spreading of the disease. If relativity more than 70%, which is coupled with warm weather, that is the temperature range between 12 to 25 centigrade, and intermittent trends helps in development of the disease. So now coming to the management. First, since it is seed-borne disease, therefore the first management will be seed treatment with a fungicide. Actually, there are so many fungicides available in market which is taken as a seed treatment. But for management of Alternaria blight of mustard, seed treatment should be done with the help of Ridomil MZ at the rate of 2 gram per kg seed. Second one, cultural management. First, we will use resistant variety of the seed. Second, next, timely sowing. Timely sowing is also very important. If we will sow the crop in time, then definitely there will be less chances of appearance of the disease. So the timely sown date will be 10 to 30 October of every year. So timely sown is very important for management point of view. So next one is proper field sanitation. Field sanitation, because the pathogen also survives in crop debris also. So field sanitation is also important for management point of view. timely weeding is also important because the pathogen also survives in weed also. So removal of weeds or collaterals from the field is also very important. Next, application of potassic fertilizer. As we know, if you apply potassic fertilizer in recommended dose, then definitely we can manage the disease. Next one will be chemical management. Chemical management is first spray mancozeb at the rate of 0.2% or maybe ridomil MZ at the rate of 0.15%. When we talk about ridomil MZ, then definitely we can say it is actually a combi product of metallixin and mancozeb, which generally we use 400 grams in 250 liter of water per acre. So if 1.5 gram ridomil MZ will 1.5 gram per liter of water, if it is sprayed ridomil MZ, then definitely we can manage the disease easily. Next one disease is white rust. White rust is also very important disease for mustard crop. So the causal organism is Albugo candida. Earlier it was known as Cystopus candidus. Actually, albugo is obligate parasite. Before going in detail, first of all, I would like to obligate parasite. Generally, parasite is two types. First one is obligate and second one is facultative. Obligate parasite means 100% parasite, and facultative parasite means actually it is saprophyte, but in certain condition they behave like a saprophyte. So that is called obligate. That will be the facultative parasite. Facultative parasite means actually it is saprophyte, but at a certain condition they behave like a parasite. That is called facultative parasite. So, but Albugo candida is an obligate parasite. So, we can say obligate parasite cannot be cultured in artificial media. Artificial media means PDA, like oatmeal extract, and so many medias are available. But generally, we have for isolation of pathogen or for maintaining of pathogen, we use protodextro agar media that is called PDA. So, Albugo candida we cannot cultured in artificial media. So before going symptoms and disease cycle, first of all I would like to say about classification of the fungus. That the kingdom is Mycota, division is Mycota, class Phycomycetes or Mycetes, and order is Pernosporales and family is Albuginaceae. This is the actually classification of the fungus. Now coming to the symptoms. Actually, the symptoms of the disease is generally local and systemic infection, which is observed in the plant. When we talk about a systemic infection, then in case of local lesion, in case of local infection, white creamy yellow raised pustules appear on the leaves, which later coalesce to form patches. In systemic infection and during humid weather, mixed infection of white rust and downy mildew cause swelling and distortion of the stem and floral parts due to hypertrophy and hyperplasia and develop staggered structure. Here there are two terms. One is hypertrophy, second one is hyperplasia. Hypertrophy means in both cases the size of the cell is increased. If size of the cell increase due to number of cell, that is called hyperplasia. If size of the cell is increased due to size of the cell, that is called hypertrophy. So two terms are very important: hypertrophy and hyperplasia. Hyperplasia means if size of the cell is increased due to number of cell, that is it is called hyperplasia. If hypertrophy means the size of the cell is increased due to size of the cell, then it is called hypertrophy. So this is the symptoms which is showing in the slide. Generally, white powdery substance patches appears in a lower surface of the leaf. When it comes in flowering stage, then definitely at flowering stage shoots are deformed and bear a malformed flower. That is also a characteristic feature of white rust. So here clearly it is seeing malformed flowers. Next, this is the photographs of electron microscope photograph of the pathogen. Here the conidia are arranged in a chain, that is in basipetal manner. Basipetal means the base is bigger and upper portion is smaller. So it is the characteristic feature of albugo. So conidia arranged in a chain which is in basipetal manner. Now, actually albugo candida is an obligate parasite. Mycelium is intercellular, producing knob-shaped haustoria, which is seen in photographs also. You can see. So this is the knob-shaped structure, which is generally seen in the slides. So knob-shaped haustoria in the host cells are present. Each sporangia has a 4 to 8 zoospores. Survival and spread. Now coming to the survival and spread, the pathogen survives through zoospores in affected host tissues and soil. Second infection is carried out by sporangia and zoospore, which is produced new infection. So, as we have already discussed about sporangia, spores are arranged in a basipetal manner. Now coming to the weather condition. Generally, moist weather, that is relativity more than 70%, which is coupled with warm weather, that is 12 to 25 degrees centigrade, and intermittent rain helps in development of the disease. So now come to the management of the white rust. Since it is also seed-borne disease, so seed treatment is also important from management point of view. So seed treatment with freshly prepared garlic, bulb-basted at the rate of 2%, or Ridomil MZ 2 gram per kg seed is, we suggest. The second one is cultural management. Cultural management is first growing of resistant variety, timely sowing of the crop, third proper field sanitation, fourth is crop rotation. Since this is also soil-borne in nature, so therefore crop rotation is very important for management point of view. So the next one is destruction of crop debris, particularly staghead stage. This is also very important. Staggered stage. Generally, in this case, generally flowers are deformed and grain setting do not occurs properly. So there is a more and more chances of decreasing yield of the crop. So destruction of crop debris, particularly staggered of the previous year crop. Next, avoid over irrigation and or you to say avoid water stagnation in the field. Apply potassic fertilizer. That is also important for management of field. Management point of view, if we apply recommended of potassic fertilizer, then there is a more chances for management of this disease. Next one is chemical management. Chemical management. First, since this disease is caused by Oomycetes fungus. Oomycetes fungus can be controlled with the help of metallic seal. So we also recommend for management of this disease is spraying of metallic seal, or we can say metallic seal plus mancozeb, that is Ridomil MZ or Ridomil Gold. So second one, when we apply Ridomil Gold, then the dose will be 600 gram in 250 liter of water per acre. When we apply metallic seal 8% and mancozeb 64%, that is Ridomil, that is called Ridomil MZ. So we will apply 400 gram in 250 liter of water per acre. Next important disease is downy mildew of mustard. Downy mildew of mustard is caused by Pernospora parasitica. Actually, the classification of the fungus, the kingdom is Mycota, division is Oomycota, subdivision is Deuteromycotina, class Phycomycetea, Oomycetes, and order Pernosporales, family Pernosporaceae. Actually, the symptoms of the disease generally appear in leaves. In disease affected plants, yellow irregular spots appear on the upper surface of the leaf and white growth is visible on the upper surface of spots. If attack is severe, then inflorescence is also affected. The affected inflorescence is malformed, twisted and covered with a white powder called stag head. Stag head is the characteristic feature of downy mildew. No pods are formed if the symptoms of the disease appear in mustard. So we can say no pods are produced on such inflorescence. The symptoms of the disease appears in leaf as well as in flower can be seen in the slide. The fungus when attack the young seedlings, discoloration occurs and in severe cases the whole plant perishes. Purplish leaf spots or yellowish brown spots on the upper surface of the leaf appear, while fluffy downy fungus growth is found on the lower surface of the leaf. Regarding pathogen Pernospora parasitica is an obligate parasite. It has large finger sepate or clavate and branched haustoria. Condylophores are erect and dichotomously branched. Stigmata are long, slender and pointed. A single conidium is borne at the tip of each branch. Conidium are broadly oval, ellipsoidal, hyaline. Oogonia is spherical and hyaline. Oospores are globose and yellow in color. Mode of spread and survival. The fungus attack broccoli, cabbage, cauliflower. There are so many cruciferous fungus, all cruciferous crops which are affected by this pathogen. The fungus penetrates in the soil through oospores in roots or in old diseased plant parts. As contaminated with seeds, it also persists in perennial hosts. Secondary spread of the disease is through water and wind-borne conidia. Now coming to the disease cycle, the infection occurs when the soil-borne resting structure called oospore germinates and produced sporangia under moist and cool condition. The sporangia are produced under the leaves. In evening are released during day as leaves die. The wind-borne blown sporangia land on leaves and directly penetrates the leaves and flowers. The pathogen survives between cold crops in the soil as oospores. Now coming to the survival and spread. The pathogen survives as oospores, that is called resting structure, on the affected plant tissues in the soil and on weed hosts. Actually, this is soil-borne pathogen, so seedling may be killed or develop dark brown vascular systems from severe infections. But older plants are rarely killed. The disease can be devastating under cool and moist weather condition. When we talk about favorable condition, then definitely we can say temperature and relativity play an important role for development of the disease. The temperature ranges from 10 to 20 degrees centigrade and relativity more than 90% favors the development of the disease. Now coming to the management. Actually, this is seed-borne disease. So seed treatment with metallic seal, that is called Apron 35 SD. That at the rate of 1 kg per kg seed, or Ridomil MZ at the rate of 2 gram per kg seed is important for management point of view. Next one is cultural management. First, growing of resistant variety. Second one, timely sowing of the crop. Third, field sanitation. Next, crop rotation, and crop debris should be removed from the field at the time of sowing, or particularly stag heads of previous year crop. Next, avoid over irrigation, or we can say water stagnation, and application of potassic fertilizer. Potassic fertilizer means recommended potassic fertilizer that helps in management of the disease. When we talk about chemical management, then definitely we can say spray Ridomil MZ at the rate of 400 gram per 250 liter of water per acre is also helpful for management of the disease. Spray metallic seal 35%, that is called Apron 35 SD at the rate of 250 gram per 250 liter of water. It means 1 gram per liter of water. That is the recommended dose of Apron 35 SD. And now coming to the next important disease, that is Sclerotinia stem rot of mustard. This disease is very important from a cultivation point of view. So actually this disease come in Jharkhand in very few, but in case of Bihar, in case of Uttar Pradesh, this disease is very common in mustard field also. This disease is caused by Sclerotinia sclerotium. The symptoms of the disease generally appear in leaf, which is generally appears as a elongated water-soaked lesions, appear on stem also, near to the crown region, covered with cottony mycelium growth later on. Plant look like a whitish from distance at internodes or base. As the disease progresses, the affected portion of the stem develop a bleached appearance, and eventually the tissue thread girdling of the stem results in premature ripening and in lodging of plants. Hard black bodies, the sclerotia, are formed inside the stem, and occasionally on the stem surface basal stalk infection rarely occur. This is the photograph of the sclerotia rot. Generally white patches are appearing in stem also, which is clearly seen in the photograph. The next is disease cycle. The disease occurs when soil-borne sclerotia, that is dormant, or we can say resting stage, germinate when the soil surface is continuously wet for at least 2 weeks. The germinating sclerotia from small 10-cup separate structure called apothecia, which release millions of airborne sexual spores, that is ascospores, that colonized dead plant parts such as senescent flowers and infect plant tissues. The sclerotia can survive in soil up to 8 to 10 years. So the disease is most destructive during cool and wet weather in dense plant. Now coming to the survival and spread. The pathogen survives as a mycelium in dead or live plants, and as sclerotia in infected parts, or on soil surface with seed as contaminant. Favorable condition, actually high humidity and temperature, and wind current also play an important role for development of the disease. If humidity is more than 90 to 95% and average temperature is 18 to 25 degrees centigrade, as well as along with wind current, also helps in development of the disease. So from management point of view, if disease appears, then it is very important for management. So management point of view, first of all, since it is soil-borne disease, so deep summer plowing is very important for management of the disease. Second, disease-free seeds or resistant varieties should be grown. Third one, since it is soil-borne, this pathogen survives up to 8 to 10 years. Therefore, for at least 4 to 5 year crop rotation is very important with non-host plants, so that the disease can be managed. Field sanitation is also important for management point of view. Avoid dense planting so that the plant can be grow properly. In this way, the disease can be managed easily. This is all about the mustard diseases. I think 4 diseases we have covered in this topic. The next crop disease will be discussed in next lecture. Thank you.